Hey folks, Miss Psilos here to teach you about the different pieces of evidence used to support the theory of evolution. Theories and ideas of how organisms change over time began in the early 1800s, but it wasn't until 1859 when Darwin published his book on the origin of the species for evolutionary theory to become revolutionary and controversial. Since then, scientists have been collecting samples of living organisms and noticing patterns among them to explain evolutionary relatedness. Starting from prokaryotes, the simplest living organisms on Earth, to the most complex, how did we get where we are today? What evolutionary progression took place, and what mechanisms explain that process? So, today we will look at the major categories of evidence used to support and explain the theory of evolution. To start, I'll give you a brief recap of Darwin's travels to the Galapagos that led to the ideas that he proposed in his book on the origin of the species. This portion of the video is not something that you're going to need to take notes on, as it likely should be a review of information that you've heard of at some point in the past. We'll then start looking at our categories of evidence for evolution, first being anatomical evidence. The second category of evidence will be fossil evidence, and the third and fourth will be developmental and genetic evidence, respectively. Make sure that you take detailed notes during this lecture, and if your teacher has provided you a note-taking guide, to follow along with that as well. Let's start with Darwin. Now, I want to preface this by saying that Darwin was not the only evolutionary biologist to study the theory of evolution. There were many people before him proposing ideas like Jean-Baptiste Lamarck and Alfred Wallace. Darwin's theory piggybacked on some of these ideas proposed before him, but he put his own spin on evolution and was able to accurately detail and put forth the mechanism of natural selection to explain how it was possible for evolution to occur. Okay, so in 1832, a 22-year-old Charles Darwin was assigned to board the HMS Beagle to work as a naturalist on a journey to South America. The idea was to survey the coasts of South America to see what was out there. As a naturalist, Darwin was responsible for taking note of the landscape of every area that they visited and collect samples of living organisms endemic to those regions. This information would help them get a better understanding of the area and fill out the map of South America's shores. It was during this time when Darwin began to take note of some interesting things regarding living organisms. While in Argentina, he came across fossils of organisms that he had never seen before in any book he studied or in person. Although they seemed to possess physical characteristics that looked similar to animals living at his time, for example, if we look at the top organism, it reminded him of an armadillo or a turtle because it had a hard carapace on its back that shielded its back. And if we also look at this one down here, it had claws like a sloth. But these fossils that he found demonstrated that these organisms were ginormous. They were a lot larger than any organism that they might be related to. So from these fossils, he realized that there must have been living organisms that were different that lived in the past, different from those species that live today or during his time. He took the fossils with him and moved on. The next important location was off the coast, the west coast of Ecuador. The voyage stopped at the Galapagos Islands, and it would be here where Darwin would understand and develop the most important piece of his theory of evolution. While in the Galapagos, Darwin noticed some interesting things between living organisms on different islands. There were giant tortoises, finches, marine iguanas, and penguins. These species had unique features compared to any other species he saw in South America and even around the world. He believed that they were endemic, meaning that they were only found in the Galapagos Islands. Not only this, but as he went around to the different islands, he noticed small differences among the populations of each species. For example, in the giant tortoises on Hood Island, he saw that the tortoises here had elongated necks and flat saddle-like shells. But on Isabella Island, the tortoises had a dome-shaped shell with much smaller necks. With the finches, he saw that on some islands there were dark colored finches, while on other islands they were lighter in color. They all varied in beak size and shape, and noticed that they varied in what they ate on the islands or where they made their nests. Why would there be such a difference if they were all part of the same species? Were they actually different species of very similar organisms? But why would that all be the case? He began thinking of answers, he collected his samples, and then moved on. Once he returned from his trip, he compiled all of his data, along with other available data, and came to the following conclusions. Number one, there must have been an original organism, an ancestor, that gave rise to all the variations he observed on the different islands of the Galapagos and in other regions that he visited. Number two, over time, new variations emerged, and the variations that each possessed were connected to their environment. And three, the characteristics that each organism possessed helped them survive in their unique environment. And over time, that would mean that the variations between populations allowed for the creation of new species. So he wrote his book on the origin of the species and published it in 1859, detailing the process of natural selection and how it would ultimately create new species over longer periods of time. He drew out his version of the evolutionary tree, starting with the ancestor at the base, where you see it's circled number one, that would give rise to new species that existed on the branches. Each branching point represents descendants who had modified characteristics from the ancestor, who changed in some way over time. At the tips, we reach the living organisms today and what traits that they might have inherited from those ancestors. The ideas in his book were both revolutionary and controversial, as they directly countered many ideas of theology at the time. As a result of the popularization of the evolutionary biology, other biologists were inspired to find evidence that would support or show validity to Darwin's theory of natural selection and evolution. Today, we find there are many pieces of evidence that do support the theory of evolution, and those pieces of evidence can be categorized into the following: anatomical, fossil, developmental, and genetic. Out of all of these categories, genetic evidence is the most concrete and most reliable piece of evidence. But let's get into anatomical evidence first. What counts as anatomical evidence? If we're observing and comparing physical body structure similarities among organisms and even among species, then we're considering anatomical evidence. As the name suggests, you must be looking at anatomy, internal and external. If we find that the makeup of certain body parts originate and are formed from the same structures, we would say that there is anatomical evidence for relatedness between species. For example, take a look at the forelimbs of these four different species of animals. The bones are what we're comparing among these species, their sizes, their shapes, and patterns of connection. Although these forelimbs are used for different purposes, different functions, their makeup or their structure is exactly the same. For one, they're all made of bone. They all also start with an elongated humerus that connects to two long bones, the ulna and the radius, that then connect to a series of small carpal bones, which then connect to thin but longer metacarpals and end with phalanges or the finger bones. They all have variations of these five finger bones, with an exception to the whale, which was determined to have fused with some other bones. But on the other hand, and no pun intended here, if we look over at our scales, shell, feathers, and nails, these two serve as anatomical evidence for evolutionary relatedness. All these structures are made in most part of the same material, keratin, and serve the purpose of protection in some way. Why would they all possess such similarities in these features? According to evolutionary theory, they all inherited these from an ancestor in the past. If so, the body structures that are theorized to have been inherited from a recent common ancestor would be called homologous structures. Homologous structures are the same anatomically. They are made of the same material and composed or formed in a very similar fashion. However, homologous structures might be used for different functions. If we look at our arm bone example again, the human uses their arms for grabbing, the cat for walking, the whale for swimming, and the bat for flying. But all of these arm bones are the same. So, if species evolved from the same ancestor, that ancestor should have passed on the same anatomical features to the descendants. And so, if the human, cat, whale, and bat all descended from the same common ancestor, it makes sense why they all possess the same arm bones connected in the same pattern. Here are some examples of homologous structures in some other animals. One reason why we group particular animals into the category of mammals is because they all share specific homologous structures. All mammals have fur, 3 ossicles or ear bones, mammary glands that make milk, and a uterus which grows their young within a placenta and will be birthed live. Because they all possess these homologous structures, they likely inherited them from the same ancestor in the past. Here are some more, and this time we'll take a look at a more varied group of animals. Here we see an evolutionary tree called a cladogram with only a few species represented. On this cladogram, we can see jaws evolved at this point in time right there, if you can see my mouse. That implies that everyone who evolved after this point in time should have inherited jaws from this ancestor. So jaws would be a homologous structure for the perch, salamander, lizard, pigeon, mouse, and chimp. We could use that same understanding for any of the other homologous structures listed on this cladogram. Now, here's a different piece of anatomical evidence used to support the theory of evolution. They're called vestigial structures. These are body structures that are no longer used by the individual, but they're still present in their body. Then why have these body parts still if they're not used? The theory is that in the past, an ancestor did use these body structures, but over time, they lost function in their descendants. The fact that they're still in the bodies of the descendants supports that they were inherited from a common ancestor. In humans, there are 3 apparent vestigial structures. On our skin, we have erector pili muscles that make our hair stand on its end. You've experienced this whenever you get goosebumps. In other animals that have much more hair than we do, making hair stand up is a defensive mechanism to make oneself look larger or intimidating. It also is used to conserve heat if it's cold. While in the modern human, neither of these things are necessary, and yet it still happens. Additionally, we have an appendix that houses bacteria, but is largely unnecessary as it serves no digestive function anymore. And we also have a tailbone, yet we have no tail. Why have these things? Likely because an ancestor of ours used these for a purpose, but that purpose was lost over time. Still, these structures were passed on and are still found in us today. We inherited them from a previous ancestor. Here are some other examples of vestigial structures in other organisms. In snakes, which don't have arms or legs, we still find bones for their hind legs hiding internally, indicating that they likely descended from an ancestor which used to walk. We find similar things in whales, which have an extremely reduced pelvis and leg bones that are hiding inside them. Penguins have flippers that are much like the wings of other birds, but they don't help them fly. These flippers still have feathers on them, though. Now, anatomical evidence is great because it easily allows us to start grouping species together that share homologous structures. But unfortunately, anatomical evidence is not extremely reliable. Here's why. Sometimes homologous structures are not always obvious. Take a look at the beaver and the elephant. The front teeth of the beaver and the tusks of the elephant are actually the same teeth. They're both incisors. But looking at their appearance, that's not what you'd think right off the bat. It's not extremely obvious that they're actually homologous. This happens quite a lot. Additionally, some physical structures can trick you into thinking that they are homologous when they actually aren't. Take these streamlined bodies of the shark and the dolphin. Both are adapted to living in an aquatic environment to swiftly cut through the water at high speeds. They have fins on their back and smooth bodies. They have holes that allow them to breathe as well. You'd think that these 2 organisms, these 2 species, are closely related and inherited these features from the same recent ancestor, but that would be false. They evolved these features separately at 2 different points in time from 2 different ancestors. Instances like this can really trick evolutionary biologists into grouping organisms together on an evolutionary tree that shouldn't be. This is mainly why anatomical evidence is not extremely reliable if it's used alone to explain evolutionary relationships. These tricky looking structures are actually called analogous structures. These are body structures not inherited from the same ancestor. They evolved at two different points in time from two different ancestors. Analogous structures may be used for similar purposes or similar functions, but deep down, their structural makeup is not the same. In dolphins, their skeletons are made of bone, while in sharks, it's cartilage. Sharks have gills to directly breathe oxygen from the water, while dolphins need to go to the surface to get oxygen from the air. Dolphins are more related to mammals since they have mammary glands, hair, and give birth to live young. But their streamlined shape and smooth body, though those are adaptations to their aquatic lifestyle. And this is why they resemble sharks externally. We can see from this cladogram that the streamlined body of dolphins evolved at different points in time compared to the sharks, much later. And that streamlined body was inherited from an ancestor who returned to aquatic life, sorry, after living on land for some time. So you can see here's the ancestor of the sharks, and here's the ancestor of the whales and dolphins, the cetaceans. Two different ancestors evolving the streamlined body, two different points in time. So these are analogous structures.
Alright, so that's anatomical evidence. We'll now move on to fossil evidence for evolution. Starting out simple, what's a fossil? A fossil is the remains or an impression of an organism that has been preserved within a mold or within rock for hundreds to thousands to millions of years. On the screen, you can see that there are multiple different types of fossils. It's not always bones that we find, it could be an impression of a body structure, and in some cases, that impression is filled in by sediment and therefore becomes a cast of that structure. Other times, like in a lot of cases with ancient insects, their entire body might be encased with an amber, which is solidified tree sap. Fossils can show us a lot of important things. In general, the fact that there are fossils of organisms that don't exist today tell us that life is old and has changed progressively and or dramatically over time. Fossils can also show us who the ancestor of organisms living today might have been. They could also fill in the gaps in an evolutionary tree and serve as transition organisms. Transition fossils show us small changes in body structure that allow us to visualize progression, how ancestor A became living organism B. Take human evolution, for example. Evolutionary biologists and anthropologists continue to find more evidence of human like fossils that allow us to see how we may have evolved from an ancestor that we share in common with chimpanzees. Each hominid fossil found serves as another transition point from that ancestor to the modern day human. We can see changes in posture, teeth, arm and leg length, and skull size that may help to explain how our modern human features evolved in small progressive ways over time. As more and more fossil evidence is found, the fossil record continues to grow and fill out. The fossil record is the history of life on Earth as documented by fossils. Will the fossil record be completed ever? Not likely, because not all organisms that lived in the past will have fossilized in the first place, and some fossils that may have formed could have been destroyed as the Earth changed or was modified. Who knows if we'll actually be lucky enough to find all the fossils out there too? But still, as evolutionary biologists continue to add more fossils to the fossil record, we can learn these important things. We can learn points in time when particular groups of living things evolved. We can see the progressive changes in traits and structures of living organisms over that time period. It also gives us an idea of the climate and the environmental conditions that a fossil or an organism that gave rise to that fossil might have lived in or might have been exposed to. And of course, what characteristics allowed species to move from environment to environment. One big transition point is from aquatic life to terrestrial life. So, how do we know how old a fossil is? The main method used is radiocarbon dating. In this method, scientists measure the amount of carbon isotopes that are found in a sample. One carbon isotope, which is a radioactive isotope, and it's called carbon 14, it's unstable, and it'll decay at a known rate to become nitrogen 14. Since there is carbon in soil and in living structures, scientists can measure the rate of decay of this isotope to give a relative age of a layer of soil or to a fossil directly. So to figure out the age of a fossil, scientists can look at two things. The first is to observe the layer of Earth that the fossil was found in. Layers of Earth are carbon dated, and the further down the layer is, the older it is. So the fossil record, so if the fossil was found deep in, let's say, this dark gray layer, and let's say this layer of Earth is 1 million years old, then the organism that this fossil belongs to would have lived on Earth 1 million years ago. Does that make sense? The second is that if the bone fossils themselves are found, radiocarbon dating can be directly applied to the bones to determine how old they are exactly. Okay, so now we're going to move on to the final two pieces of evidence for evolution. Developmental evidence involves comparisons of developmental progression between species and their anatomical similarities as they progress through their stages of development. All life begins as a single cell. If we're dealing with a multicellular organism, that single cell will multiply trillions of times to become a fully developed and functional living thing. As the living thing develops, it will grow particular body parts in a particular order. It may have certain genes expressed and proteins active at certain times during those developmental stages that influence the manner in which their parts grow. If we compare this developmental progression between living organisms, similarities can be found. We would say that homologous structures can be seen in the embryos of vastly different organisms, indicating that they inherited similar developmental processes and stages from the same ancestor, and therefore they are related to each other. Two apparent pieces of developmental evidence are the following. In embryos of animals, there's a structure called a pharyngeal slit, or sometimes called pharyngeal arches, that develop early on. In early embryos of fish, reptiles, birds, and humans, four vastly different animals, these pharyngeal slits develop in the exact same way, and around the exact same time. Although in later stages of embryonic development, the pharyngeal slits will change to form different structures. In fish, they'll develop into the arches that form their gills, and in humans, they develop the bones of our jaw, neck, and ear. But the fact that they're all present and the same in early embryo stages indicates that we inherited this developmental structure and pattern from the same ancestor. Additionally, there are similarities in genes that are activated during embryonic development. These are called Hox genes, and they're responsible for developing body plans in animals, meaning they decide where the head goes versus the butt, where the limbs grow, and so on. From fruit flies to humans, scientists have seen that Hox genes are passed on through the evolutionary tree. More complex species may have more Hox genes than others, but the interesting thing is that the pattern of activation of these Hox genes is extremely similar among the different species. Last but not least, there's genetic evidence for evolution, and this is the most reliable and concrete evidence there is. All living things possess DNA, and what makes up that DNA is the same across the board. The use of that DNA is also the same. Genes code for proteins, and proteins are composed of amino acids. Those proteins will perform specific functions that will give cells unique abilities if they express the genes that make those proteins. If we compare the genes that living organisms possess and compare the structure of their proteins that those genes are making, we find that there are similarities, then we have genetic evidence for evolutionary relationships. Related species should have inherited genes from the ancestors they have in common. Here's what I mean. Take a look at the image on the left. Here we're comparing the DNA sequences of 23 species for the same gene. The colored areas are the areas of the DNA sequences that are the same among all 23 species. The areas that are not highlighted show areas of the sequence that vary or are different among the species, and the areas that are boxed in red may be spots in the sequence that demonstrate evolutionary relatedness. A higher degree in sequence similarity suggests that those species are more closely related. We could argue the same thing for amino acid sequences, since those are determined by the DNA. Any changes in amino acid sequence or protein sequence are the result in changes of genes. So again, the more similarities in amino acid sequence that we see among different species, the more closely related those species are likely to be to each other. When it comes to genetic evidence, the largest piece of evidence to support evolutionary theory are these highly conserved genes seen in the DNA of even distantly related species. Highly conserved genes are those that have remained relatively unchanged through time. meaning that very few mutations have occurred within these genes over millions of years. Their mutation rate is very low. This suggests that these genes are necessary for basic functioning for life. For example, we have some genes in common with bacteria, although the sequences of these genes are not exactly identical. The small differences and the amount of differences between the sequences of highly conserved genes can show us how long ago something diverged from an ancestor and how closely related they may be on an evolutionary tree. Take the histone gene and protein. This protein wraps the DNA to tightly coil it, which is especially necessary to form chromosomes, that chromosome shape, during cell division. This protein is made by all eukaryotic organisms, so plants, animals, fungi, and protists make histones in their cells, and they all serve the same function, to wrap the DNA. Eukaryotic organisms were thought to have evolved around 2 billion years ago. Histone proteins come from a highly conserved gene that has not changed dramatically since then, and we can compare the amino acids that build these proteins among living organisms today. Similar mutations or differences in the amino acid sequence can tell us or allow us to see the evolutionary relatedness among plants, animals, fungi, and protists. Another example of highly conserved genes are those Hox genes in animals that I mentioned not too long ago. All right, so we're going to end this video here.